Hello, and welcome to Week 3, Lesson 2, where we're going to discuss cofactors, pages 55 to 65 of the reading. We're going to look at three main types of cofactors: hemes, which we will focus on the use of hemes in both hemoglobin and cytochrome C. A heme is a planar 4-nitrogen ligand that holds very tightly to a transition metal. Then we're going to look at iron-sulfur clusters, and these are, as their name implies, compounds that contain iron and sulfur. These, we'll look at their structures and think about their use in electron transfer reactions. We will see specific examples of how they're used for electron transfer in the nitrogenase case study in 2 weeks. Finally, we'll look at zinc fingers, which are DNA recognition proteins, and we'll think about how the structure can help with their role in DNA identification. Okay, the heme cofactor consists of an iron that is situated inside of a porphyrin ring. This is a porphyrin. It is a 4-nitrogen containing ring. The iron sits directly in the center here, and this ring consists of 4n plus 2, an 18 pi electron system. This double bond and this double bond are not in the system. It's an aromatic ring, so it's a rigorously planar molecule. So this is the parent porphine, the parent porphyrin molecule. It's extremely insoluble, and it's not used really in biology. So instead, nature dangles on a variety of functional groups. So this is actually the porphyrin that is used in heme. This is known as protoporphyrin 9, and you can see that there are a variety of functional groups that hang off of the outside, which help it to remain soluble and can be brought into the protein itself. A more synthetically relevant porphyrin is this one. This is known as tetraphenyl porphyrin, and this porphyrin is used to model the behavior of the naturally occurring porphyrins. So if you think about the steric requirements of 4 nitrogens in a macrocycle like this, they're pretty strict. You don't have a lot of structural variability of the iron center in the heme. You can certainly have different electronics for these different groups that are substituted, but you're not going to have very different sterics. The actual structure of heme, however, can be changed by having different protein side chains. So this is a methionine and a histidine. So this is what's found in cytochrome C. And then if you have 2 histidines, that's what's found in cytochrome B. And the axial ligands actually can change the redox potential pretty substantially, as well as the protein environment that's present surrounding the iron center. Hemoglobin is another protein that contains a heme group. So in that structure, one of the axial ligands is missing. That's where the oxygen binds. We'll talk more about oxygen transfer in the next lecture later this week. One last thing to note about these heme proteins is that they're highly colored. So this is a UV-Vis sample of both oxidized and reduced cytochrome C. I couldn't find a picture of the actual solid sample, but on the next slide I do have some examples of some solid samples of similar molecules showing their really bright colors. The main takeaway I'd like you to get from this discussion of heme is the structure, that you've got this planar nitrogen macrocycle with different ligating groups axial to the porphyrin ring, and that those axial groups and the protein environment can cause significant redox potential changes that allow electron transfer reactions to take place. In addition to heme, there are other highly colored centers in biology using a chlorin or a chorin ring. So these are slight modifications to the porphyrin ring. So this is the chlorin complex found in chlorophyll. You can see that this is a fused ring system, and so that differs slightly from the porphyrin ring system. And chlorophyll has these bright green colors. And then over here we have a cobalt chorin complex. So this, again, it's not exactly a porphyrin. It's been slightly modified. This has a cobalt in it. This is coenzyme B12, which is this really brilliant red color. And we'll talk more about coenzyme B12 in lecture 4 this week when we talk about carbon group transfer. And there are some aspects of photosynthesis that are discussed in the book chapter, but we're not going to be talking about it in class. Let's now move on to the iron-sulfur clusters, which are very common redox centers in biology. This first example is a rubidoxin. It's an iron surrounded by 4 cysteines. These are actually only found in bacteria and very low life forms, not higher life forms. Iron-sulfur clusters in general are usually found close to the surface of a protein. The electron transport from one protein to another. Electrons move from the surface of one protein, an iron-sulfur cluster on the surface of one protein, to an iron-sulfur cluster on the surface of a second protein. And since electron transport falls off very strongly with radius, these tend to be found relatively close to the outside of the protein to allow that electron transfer to take place. The highly variable redox potential in rubidoxins is due to the exact protein environment. So side chains over here, that can change the polarity of the substrate, or of the pocket, can change the redox potential. I've been talking in the homework about, and also I mentioned it in one of the earlier videos, these consensus binding sequences. This is the DNA code, so it's the amino acid code for the binding of the iron by the rubidoxin protein. So we have a cysteine, a proline, an X amino acid, then another cysteine, a glycine, some variable number of X amino acids, a cysteine, 2 X's, and a cysteine. And so one of the homework questions asks you to think about why these sequences are conserved across biology. And the textbook goes into this in a little bit of detail. The ferredoxins is the next class of iron-sulfur clusters. And these are variable. They can have 2 iron, 2 sulfur, 3 iron, 4 sulfur, or 4 iron, 4 sulfur. They're all bound into the protein with cysteine residues. And these are found across life forms from bacteria up to mammals. And they have a large structural diversity and a large redox diversity, again, due to the exact environment of the protein. But they have a pretty common consensus sequence that is pretty similar to what we saw in the rubidoxin. So cysteine, some number of X amino acids, another cysteine, XX, cysteine, X, and cysteine. So these cysteines need to be in a conserved location so that they can get the iron bound in the appropriate place, and the variable amino acids can tune the redox center. One special class of the ferredoxin is the high PIP, which is the high potential iron-sulfur protein. These have the same binding. They look exactly the same as the regular ferredoxin, but have a redox potential that's 800 to 820 millivolts higher. So why is this? And the reason is the exact amino acid nearby, the amino acids nearby this ferredoxin are very different in the high PIP iron centers. The higher oxidation state in the high PIP center actually is pretty unstable, and so there's some nearby hydrophobic side chains that allow this hydrolytically sensitive species to exist. It turns out in modern forms of life, so higher organisms, instead of using the high PIP iron centers, they use other types of centers, so iron-sulfur clusters, hemes, or copper proteins, which are much more relevant. Interestingly, copper proteins only came about after the photosynthetic extinction event that made iron 3 much more common and therefore much less available, since iron 3 is rust in the environment. So there's going to be more discussion of iron uptake and storage and the importance of iron that predates photosynthesis. And so our core proteins still use the iron too, because that's how we evolved, but we don't have ready access to iron 2 in the environment anymore. Makes it very difficult for living things to take up iron, but also very important. The final protein is the Rieske iron center, which has cysteine and histidine binding sites, and so there's the consensus sequence there. And so you can imagine that having both sulfur and nitrogen donors is going to change the redox potential. I'm not going to ask you to know how these different centers have different redox potentials, but I'm going to want you to understand that different amino acids and different protein environments do cause a significant change in redox center. These Rieske centers are found in both eukaryotes and prokaryotes, and they allow for sort of a higher oxidation state redox activity to take place. The final cofactor that we'll consider are called zinc fingers. These are really cool little proteins that allow for DNA recognition. So anytime you need a protein to interact with DNA, it's likely that you've got a zinc finger going on to help that take place. So this is a very small little protein. There's a zinc here. It's bound by 2 histidines and 2 cysteines. There's some beta sheets and an alpha helix, and that's the whole protein. So often the proteins are linked together, several at a time, and then this whole unit is then attached somewhere down here to a larger protein, let's say a DNA transcription factor, that wants to bind to a specific DNA location. And you learned this in biology, presumably. I did. And so how do you get the DNA recognition? It's the amino acids on these alpha helices, the variable region of the alpha helices, that allows the DNA recognition to take place. And I found a really cool picture that I'll show on the next slide that shows that. Zinc is there to actually fold the protein together. You can imagine that if this zinc wasn't here, this beta sheet portion and the alpha helix portion are not going to be folded in the appropriate way. And so that zinc is essential for the structure of the protein. And zinc is handy because it doesn't have any redox activity. Zinc is a D10, 2+ cation. There's no real redox availability besides that. And that's what you want because you don't want to have oxidative DNA damage, which could cause mutations. If you want to learn more about that, talk to Prof. Haushalter. That's what his research area studies. The way you can get different DNA recognition is that this alpha helix, although it's important structurally, these 2 histidines have to be in these exact locations. The rest of the amino acids on this alpha helix are highly variable, and you could imagine putting good hydrogen bond acceptors and donors in there to bind to the DNA bases, and that is where you get the high degree of specificity for binding to the DNA. If you go to this website, the Protein Data Bank, you can actually explore a zinc finger protein on your own. I'm not going to expect you to know this in detail, but I wanted to at least show it to you. And so this is just a screenshot that I took when I was looking at it. And so we have the zinc and the cysteine and the histine, and the DNA binds inside these 3 zinc fingers, and the important part here is these red amino acid side chains, and you can't really tell from this picture, but that's a histidine. This is an arginine or an aspartate, I don't remember, but there's a hydrogen bond donors and acceptors that point directly into where the DNA is, and that's where you get the recognition of the DNA. And that's shown in even more close-up view on the next page. So I played around with this for a while, and if you're curious on how I got this image, then you should just talk to me. I'm not sure I could recreate it easily. But what I did is I selected this, I believe it's an arginine, there's a carboxylate. And so you can see there's a lot of hydrogen bond interactions between the DNA bases. So these are DNA bases. And so you can imagine having an arginine and a carboxylate right here, or it's going to bind differently to like a GT or a GA pair. And so this is how you can get the really strong specificity of binding between a zinc finger and DNA, and allows for very strong recognition of DNA. So homework question, one of the homework questions this week has you look at and explain in a few sentences why zinc fingers have multiple DNA binding regimes. Why do you tack 2 or 3 of these zinc fingers together when you want to have good site specificity? So think about that, write up your answer, and that'll be that. So, what are the take-home messages for cofactors? I want you to appreciate the structures of these cofactors. The iron-sulfur clusters are redox centers, the zinc fingers are DNA recognition, and hemes are important also for redox, but also oxygen carrying. So you've seen some of these structures, and we'll begin to use these structures to do chemistry starting in the next lecture, where we look at iron and copper uptake. And then lecture 4 this week is oxygen transfer and carbon transfer.